In this chapter, we are going to understand the various biotechnological techniques like gene cloning and how they are used to benefit various aspects of human life. And it is an extension to the chapter of biotechnology principles and processes. Now, basically, when we talk of biotechnology, we are essentially dealing with industrial scale production of biopharmaceuticals and biologicals using genetically modified microbes, fungi, plants, and animals. In a literal sense, it's a technology where you're using living organisms to either modify some products or synthesize new products which are ultimately beneficial for human beings. Ultimately, it is useful for human beings, and that is the very essence of biotechnology. And the applications of this science of biotechnology is seen in therapeutics, it is seen in diagnostics, it is seen in by production in agriculture, it is seen in processed food, it is seen in bioremediation, waste management, energy production, it is used in food and dairy technology, drug development, pharmaceuticals, so on and so forth. So the applications of biotechnology is widespread in various fields which are of science, and all of them ultimately are done to improve the quality of the human life. Okay, so biotechnology in agriculture, specifically biotechnological applications in agriculture, they can be used specifically for food production. See, Green Revolution happened in the 1960s. The father of Green Revolution is Norman Borlaug, and his student, Dr. M.S. Swaminathan, has obtained the Green Revolution in India. And the basic concept behind Green Revolution was to increase the yield of food production, which developed as a short due to the continuous world wars and very cold wars that were happening in that. The entire geopolitical conditions in the world at that time created a lot of food shortage and food supply deficiency, and that was the main purpose behind Green Revolution. Green Revolution, basically what it did, it used hybridization techniques, conventional plant breeding techniques, they used fertilizers, they used pesticides, herbicides, a lot of chemical products were used to increase the crop yield. Now, but today, if you look at today's world, even the Green Revolution is not enough to to increase or to come to the level of food deficiency supply that we are facing. We are again facing food shortage. A lot of countries are the population is suffering from malnutrition or undernutrition or some sort of food deficiency, essential food grain deficiencies. All this is happening due to the increase in population, famines, a lot of factors. But anyway, conventional methods are not successful as what was during the time of Green Revolution. So biotechnology can be used for increasing food production, where one, it can be used in that agrochemical based agriculture, the kind of agriculture which was completely dependent on chemical products in the form of fertilizers, herbicides, pesticides, insecticides. So in that field you can put biotechnology and improve agriculture. You can create organic culture. Now organic culture is nothing but the kind of culture where without any using any chemicals, completely naturally, by through natural methods you are producing food materials. The third one is creating the genetically engineered crops. Now genetically engineered crops are nothing but genetically modified organisms. See, conventional breeding is or plant breeding happens within the existing varieties, and no matter how much superior of an variety you have, they still have some sort of problems. So if we take a conventional variety and modify it in such a way, genetically modified in such a way that you remove the problems also which are existing in the superior variety. Let us say I have a very superior variety of wheat. It is high yielding, it produces a lot of grains, but still it has one problem: it is highly susceptible to temperature changes. If I can remove that, I can create a plant which is superior in yield and does not have that temperature change problem by genetically modifying it. then I am solving my problem. And this modification cannot be achieved by conventional breeding. It can be only achieved by genetic engineering. And what is genetic engineering? You are breaking down the barriers of genes by transferring genes of one species into another species, or by altering the existing genetic condition of an organism and produce something huge. So this is the next thing that is used, and that's why it's called as gene revolution. So genetically modified organisms are present in plants, they're present in bacteria, they're present in fungi, and they're animals. And basically what happens is their genes are getting modified, they're undergoing manipulations, they're getting altered, and hence the name genetically modified organisms. Now let us see the biotechnological applications in agriculture. Some of the applications of biotechnology, there are many. The first and most important is production of pest-resistant plants, which could decrease the amount of pesticide usage, and the best example is BT cotton. BT cotton. Now, as I have said, this comes under the agrochemical area of agriculture. So, we are targeting the agrochemicals. So what is happening here? We know that to protect our cotton plant, we use increase of amounts of pesticides. Why? See, cotton, this is a cotton bud. What you can see here is a cotton bud, and you see this bud has holes. This is due to bollworms. Bollworms, they create holes, they destroy the buds, create holes in the buds, and as a result the bud won't open. Normally if you have this kind of an opened bud, this is cotton, the cotton that you need, required, but this is completely destroyed, it is now useless. As a result, the entire yield of cotton is getting destroyed. So how do you protect the plant? We are going to spray pesticides which will kill these bollworms, and these pesticides will get into the soil, which are again harmful to the soil, and then there is a lot of chemical pollution talk that we all know is happening. So now scientists have come up with a way in which you can reduce the use of pesticides by making the plant itself resistant to bollworms. Means you're making the plant so strong, and you're giving the plant itself the necessary equipment to fight against the bollworms. It is like somebody is invading your country, okay, or your home. A burglar is coming into your house every day. You are expecting somebody from outside to come and save you. Now for that, to protect you from the burglar or from the robber, that outside persons are sitting in your home, eating your food, and destroying your resources, which is ultimately lost to you. Instead, if you get yourself ready, if you put some sticks, so if you put some locks on your home, if you put a fence around your house, then you yourself are capable of fighting the burglar or protecting your house from that burglar or the robber. Correct? So you are not dependent on others, and you are saving your resources. So the same logic existed behind it, that what if we equip the plant in a way, the cotton plant in a way, that it can fight this pest. So BT cotton actually is nothing but it contains a particular genes of a protein that is produced by a bacteria called Bacillus thuringiensis. So this protein that is coming from this bacteria is called BT toxin. Now it was found that this BT toxin acts on lepidopterans like tobacco, budworm or armyworm, coleopterans like beetles, and dipterans like flies and mosquitoes. You need to remember this. This is a direct sentence from your NCERT textbook. You need to remember these names and the examples. They're very important for the objective. So it was found that the BT toxin rather is destroying these organisms, is destroying these lepidopterans, these coleopterans and dipterans. And we know that this budworm is the main problem. The bollworms, the budworms, they are the ones that are all creating all the problem. So scientists got an idea that why don't we put the BT cotton gene in the plant? Why don't we make the plant only to produce BT toxin. So BT cotton is nothing but the plant which contains the BT gene. The gene from the Bacillus thuringiensis bacteria which is producing the toxin. So plant itself is producing the toxin which will kill these pests that are attacking it. So BC thuringiensis or Bacillus thuringiensis, the protein crystals, they are toxic insecticidal proteins. And if the plant is able to produce that, then the plant can destroy these insects, these lepidopterans, these coleopterans, these dipterans. Now, how is it destroying it? Apparently, the BT toxin is actually inactive. It is when it is produced, it is inactive and it is called as a protoxin. But when it enters into the insect, when it enters into the mid-gut of the insect, specifically the mid-gut of the insect, there the insect has alkaline pH. In that pH, this protoxin becomes active, and as a result, what it does is it creates osmotic cell lysis. It creates pores in the cells, the epithelium of the alkaline. Sorry, it creates the pores in the gut epithelium. Epithelial cells of the stomach or the mid-gut, they will have pores, they'll start to have osmosis and they will die. So in this way, it is killing the lepidopterans or the dipterans or whatever you want to take it, all those that we mentioned, right? So the activated toxin binds to the mid-gut of the epithelial cells, create pores causing swelling. How is it causing swelling? It is allowing the inflow of water, and when this too much of water enters the cell will burst, the entire mid-gut epithelium bursts out, and the organism will die. Okay, so now what they have done is, scientists have found that there were 3 sets of BT toxin genes that were present in Bacillus thuringiensis. They were 𝐵1AC, 𝐵2AB and 𝐵1AB. What are they? They are 𝐵1AC, 𝐵2AB and 𝐵1AB. Now 𝐵1AC and 𝐵2AB, Cry means what? CRY. See, it is actually CRY, which is for short for crystalline. The protoxin is in the crystalline form. That's why it is cry. Now, these 𝐵1AC and 𝐵2AB, they control the cotton bollworms, whereas the 𝐵1AB product is controlling the corn borer. Okay, so based on the crop you are using, you can use the genes. If you are using cotton, you can take the gene 𝐵1AC and 𝐵2AB. If you are using a corn plant, you will use 𝐵1AB. So based on the crop, you will take the specific gene which produces the specific toxin that targets the specific insect group. Okay, so after that, you insert the BT gene into the plant. The plant itself in its body will produce the BT toxin. When the insects attack the plant, the BT toxins enter into the insect. There, the protoxin becomes into the activated form. Activated toxin, where it is happening? In the mid-gut. And there, it will destroy the insect by killing it, without spraying a single chemical. And this particular BT toxin is no way harmful to the plant, because it is active only in the alkaline pH, and the plant itself, it will remain as a protoxin in the plant, and it's no way harmful to the plant. Okay, so this is BT cotton.